Hello, my name is Jason Linville. This lecture is being recorded for CJ 352 Forensic Science Lab 2 or something like that. It's being recorded in the spring of 2020. And this is the lecture on STR detection, also STR analysis. So we just got done with, as far as where this is at in the class, we just got done talking about amplification of DNA and then how to prepare that DNA for analysis. And we sort of covered the concepts of, okay, we're going to include a size standard in with every sample as it gets separated. We're going to run an allelic ladder in addition to the size standard in order to help convert our sizes or the time of our peaks into size and base pairs and et cetera. And I do have the waiting list turned off. But now what we're going to talk about this is, okay, after we prepared it and we know overall, okay, they're going through the capillary, and then when they hit that detector, magically there's going to be peaks. We're going to take some of the magic out of what happens at that detection window. So we're going to identify some of these things, how a dye is attached to the DNA, explain how the instrument detects it. There's a lot of sort of repetition here because the instrument detects the DNA since there's a dye attached to the DNA. And then specifically, I want you to know that the dye is excited. It just doesn't sort of fluoresce and glow. You have to zap it with a laser at the detection window. And then how is it detected? How does the instrument detect DNA? This is sort of more the overall concept. It detects DNA through a dye. And then notice that this one says how the dye is detected. Okay, and that's a camera. So all of these are pretty quick. And then we're going to go over later on sort of the problem with multiplexing, analyzing more than one locus at a time. Okay, so let's start with the detection here. All right, so how does this camera see DNA? So we're sort of jumping to the end here. The way that we're going to see DNA at the detector is not, you know, some molecular weight or hitting it with an ion or anything. We actually just physically have a camera that's looking for visible light, except the camera is super sensitive, right? So it's better than just hiring some guy to sit there and look at a small detector window and mark off when they see a fluorescent dye. But it's a CCD camera. It's just sort of like a normal camera that is detecting this light. So the camera sees DNA because, as we're going to learn, the DNA, it's labeled with a fluorescent dye. Okay, now you can amplify DNA without a fluorescent dye. You can amplify DNA by putting the DNA in a tube with primers, polymerase, and DNTPs, right? That's how we amplify DNA. But in this specific type of analysis, since we know beforehand when we do the amplification that we're going to want to separate and measure our DNA, we label our DNA in the amplification process. So actually, when we went through the process of filling out our sheet and amplifying DNA, and we had the calculation down in the bottom chart for primers, those primers were actually already labeled. When we order the primers, or if you buy primers from a commercial company in one of those kits that can amplify 16 loci at one time, the primers are already labeled. So you can, like, a primer is a fragment of DNA, okay? So you can just order that series of single-stranded letters that can serve as a primer, or when you order that series of single-stranded letters to serve as a primer, you can also say, hey, by the way, to the 5' attach a fluorescent dye to this primer. And it takes the cost of a tube of primers like from $10 up to about $80 or something, including that fluorescent dye. But still, it's pretty inexpensive because that little amount of primers go a long way. So it's relatively an easy way to see the primers and to see the amplified DNA. Okay, now it's actually just one of the two primers is labeled with a dye, but we're not going to focus too much on that in the class. Just know that the primers are labeled with a fluorescent dye. Okay, how does fluorescence work? Well, this is the same concept that we already covered when we talked about visualizing semen stains. Okay, we put high energy or low wavelength blue or ultraviolet light right onto the fluorescent stain. And the fluorescent stain had molecules that would absorb that light, get excited, and then emit light at a higher wavelength. I actually drew this kind of dumb because this is high energy down here, and then this emitting lower energy sort of up here in like the yellow or orange or whatever color goggle range it is. I forget, when did I grade those things? Was that just over the weekend? I forget what happened on Friday, but you guys should have your exam grade should be up in Canvas. That's right. And I should have mentioned, I'll mention this after. I'll just throw it in right now as well. The quizzes, I had some requests. The quiz is actually still open today, and there was a small window when the quiz should have been open, but it wasn't open. Okay, so just know if you completely forgot about the quiz, there is quiz 3. So I'll eventually release the answers to that, and we're almost caught up with all the grading and stuff. But anyhow, what made me think of that exam was everybody did pretty well on the exam when talking about the orange goggles, yellow goggles blocking that blue light. Okay. So here, the fluorescent dye that we're using, we actually order them. They're special fluorescent dye molecules. So it's not just like, let's label them with a Siemens stain or something. But they work the same way, where sort of high energy light goes in, and it's blue UV. It's also sort of, we're dealing up here in the bluish green range, okay? But it's still sort of down on that edge of the rainbow, on that end of the rainbow. And we're not going to use a flashlight. We're actually going to use a little bit more intense light source. So it's going to be a laser. Okay, so you can sort of think of this as a blue-green laser that hits the dye, and then it absorbs that energy and it releases it sort of in the red to yellow range up here. Okay, so it's the same concept of fluorescent. It's just that every primer has this fluorescent dye attached, and as the dye stays attached to the primer, which becomes part of that amplified DNA. So as your DNA is getting separated in the capillary, now all of a sudden all of our DNA also has this fluorescent dye attached to it. Okay, so how are we going to separate our DNA? What instrument are we going to use? Excuse me. We used to use a 310. We now have upgraded to a 3100. That's what happens. As the instruments get better, the numbers get bigger. Okay. But this is an instrument that separates our samples through the capillary. We talked about that. But at the excuse me, at the end of the capillary, or actually before we get to the very end of the capillary, the coating of the capillary is burned out, and there's a small clear, where you can see the clear glass tube that is the capillary. Remember, this capillary is a glass tube, very thin, about the thickness of a needle, and it's covered with a special coating that allows it to be flexible and not break. Okay, so what you do is you burn off a portion of that coating. That becomes the detector window, and it's literally just a clear tube of glass where, as the DNA goes through, any camera that's looking for any fluorescence can be seen by the camera. If you want to zap it with a laser, the laser can go through the glass and hit that fluorescent dye. Okay, so with this capillary electrophoresis, this fluorescence from the primers is being detected just with a normal camera. Okay, so you know, when we say go and are racing the DNA through the capillary, you know, we had the image of David Caruso there at the waiting line, and we replaced him with a camera at the at the finish line, waiting line. So what happens in that detector window, as the DNA starts traveling through the capillary, the laser hits the detection window. And when the laser hits the detection window, no DNA has hit the detection. So basically the camera sees nothing. The laser's hitting the detection window. Sorry, I'm dying here live on camera. And nothing's happening. After a period of time, when the DNA eventually hits that detector window, then the laser hits the DNA, it fluoresces, and the laser is able to see and say, aha, DNA is here. I see fluorescence. Okay. All right. So here we go. Why is this purple? Oh, I guess it's sort of the purpley blue laser going through, when in reality it's actually sort of more bluey green. Okay. But the camera is sitting here, and if it just sees that normal laser passing through, okay, then it's like, okay, no, that's just the laser. I'm going to ignore that, right? Okay. So the laser is what is going to detect the DNA. And early on in the process, let me get rid of these annotations here. Can I erase all ink? My red pen to confuse you. Okay, no DNA. The detection window, the fluorescent dye is going to be excited by that laser, but at the beginning, no DNA. The camera does not see any fluorescence. Okay, it's able to ignore that purple color. It knows that it's not what it's looking for. Here, this represents a fragment of DNA that has that red fluorescent dye at the end. So when the laser hits that fluorescent dye, it gets excited and it fluoresces like a red color, okay, or an orange color or a yellow color. The camera can see that fluorescent dye and it registers, oh, DNA is present, okay, and it'll sort of draw that peak, all right? Now, what we're going to find is a neat trick that we're going to get to a little bit later on is, and that 488, that's sort of for the argon laser, that's the wavelength. So remember, the blue flashlight that we had was like 450. So we're a little bit closer to the green here as far as the laser goes. Okay. So that laser is going to go in and we're going to be able to label our DNA with different colored dyes, different types of dyes. So we're actually going to be able to see different pieces of DNA in different colors. Now we're going to get more to that later on. But this just sort of shows you the mechanism. The laser goes through that detection window. If any DNA is present, the light comes off, and it actually goes through a prism before it hits the camera. I don't care that you guys understand the prism, the separation, and all that stuff. I just want you to know it's a camera that detects it, and it's a laser that excites it. And you need both of those things to see DNA. Okay, if I ask you how DNA is detected at the detection window, I don't want you to write your short answer as a camera sees the DNA. Okay, no, I want you to say a laser excites the fluorescent dye attached to the DNA. Okay, and the camera sees the dye. You answer that, what is Dr. Linville going to do? I'm going to underline the word laser, dye, camera. And then you have to ask yourself the philosophical question. What if I would have just written laser dye camera? Would I have still gotten the 3 points? Because Dr. Linville still would have just underlined those 3 words, but that's for you guys to figure out. Okay, so the last portion, we have like, damn, do I want to? I'm just going to leave this in one video, okay? I was thinking about splitting this up, but this is going to be relatively short as well, you know, if I don't ramble like I did during the first half. All right, so this portion is about the analysis of DNA, and you can see here, really this is going to be just an essay question you know is going to be coming on a quiz or exam. Explain why analyzing 2 more loci is difficult, and probably what I'm going to ask you is what are the 2 ways that forensic scientists can differentiate loci from one another? Okay, well, what are you talking? What is loci? What is different? What are you talking about? Okay. I'm going to try to explain the problem when you go from analyzing just 1 STR locus in a reaction to analyzing 3 or 6 or 16, okay? How there can be a lot of overlapping and confusion, and what the analyst does in order to help alleviate this confusion. I don't know what this is called. Okay, so this material is not in the textbook. All right. So we know all of this stuff. We know how to amplify Azure, and we just sort of learned how to measure the DNA and detect it, sort of. Okay, but we don't know how to measure many STRs. Okay, so let's go through this example that we had before. And I want you to focus on, let's say that we're just analyzing 1 locus. Okay, so we have DNA from Dr. Linville. We have my DNA in the tube. Okay, and we're going to set up a reaction with DNTPs, same for everybody, polymerase, same for everybody. Okay, and then also I'm going to pull primers that I ordered that are going to amplify TPOX. I forget do I even have a example here? No. Okay, just we'll say TPOX or whatever, 1 locus. And in my DNA, at that location in my genome, okay, I have 2 alleles, one from my father, one from my mother. One of those alleles is a 9 and one of them is a 12 repeat. Okay, so when we amplify this DNA, right, we use the primers, it attaches, it creates like 10 million copies where the allele is 9 repeats long, 10 million copies where the allele is 12 repeats long. And then we take those millions of amplified DNA, prepare them for electrophoresis, run them, okay, and we detect the peaks. Okay, now we're going to add in here this little extra step of when those, not just 1 piece of DNA, but like 10 million copies of DNA are crossing that detection window. They're all picking up that laser light. They're all fluorescing, and it's giving a very strong signal to the camera. So the camera actually sees that DNA coming across the detection window, okay? So where we're modifying it based on the lecture that we just had is, oh, it's not just the 10 million copies that are crossing the detection window. There are actually 10 million copies, and they're all labeled with a fluorescent dye, okay? That's actually how the camera sees the DNA, because this fluorescent dye is attached to every single one of those amplified copies, okay? So that's how the camera sees it. That's how we create those two peaks at 194 and 206, okay? And we know that this is a 9 and this is a 12, okay? All right. So we went over all this, right? We went over that, okay, we have the 2 times, and then the size standard, we're going to convert it to size in base pairs. We have run an allelic ladder earlier in the day, so we have this chart that we have for our allelic ladder, and we know that today on this instrument, a 194 peak is a 9 and a 206 is a 12. Dr. Linville is a 912, okay? So this is very easy to do if we're just looking at one locus, right? But now I'm going to amplify 2 loci, okay? I'm going to run the same, take my DNA, it's going to be the same, take the DNTPs, take the polymerase, set up my amplification reaction. But this time, instead of just adding one set of primers, a forward and reverse for TPOX, into the reaction, I'm going to add a forward and reverse for TPOX and a forward and reverse for VWA. Okay? Now, let's see what problems this causes. Okay, I'm a 9 and 11 and 12 for TPOX. Okay, 9 allele from my mother, 12 allele from my father, whatever. I'm a 10 and 11 from VWA, 10 allele from my mother, 10 allele from my father. Okay, I've ordered primers that are labeled with a fluorescent dye. Okay, so when I separate these out, amplify them, I get 1 million copies of this, 1 million copies of this, 10 million copies of this, 10 million copies of that, in the 10 millions range. And they're all going to migrate as I separate them through the detection window. And the detector is going to say, oh, there's a bunch of DNA. Oh, there's a bunch of DNA. Oh, there's a bunch of DNA. And this is what I'm going to get. Okay. Now, hopefully you guys can start to see the problem. Okay. I've gotten 4 peaks. I have 4 alleles, and that makes sense because I've amplified 2 loci, right? But I don't know which of those peaks is TPOX, and I don't know which of those peaks is VWA. All right. All I know is I got a peak that's 194 base pairs, 198, 202, and 206. All right. So when I go to my allelic ladder, okay, I can run an allelic ladder for TPOX. I can read an allelic ladder for VWA, and I have these charts. Okay, so I know today a 186 is a 7, a 190 is an 8, and I have these 4, but does that mean I'm a 9, 12, 10, 11? Okay, or am I a 9, 10 at TPOX? You know, because notice I still have, I'm accounting for all 4 of these. I just don't know. The point of this is I don't know which allele goes to which locus, okay? So if I want to amplify 2 loci at a time, 3 loci at a time, 16 loci at a time, I need to be creative with figuring out a way where the camera and the analyst can look at these peaks and be able to know which ones are from TPOX and which are from VWA. All right, how are we going to solve that problem? Does anybody want to look ahead in the notes or just think about, well, let's see here. Dr. Lynn, I'll give you a hint. We just amplified 4 peaks and they were all labeled with a red dye, okay? What is a way that we could differentiate the peaks? And they all came out between 186 and 210. What is a way that we could differentiate those 2 loci given that all 4 peaks are coming out labeled with a red dye between 186 and 210? Aha, Elise jumps in, different dyes. All right, and I think using the different dyes, this is going to be the easiest one to sort of visualize, okay? We order these primers, right? We can choose to have them labeled with whatever. It's not just like label them with a fluorescent dye. I don't know why we're shouting out. I rolled down my window in U-Bob. Okay, now U-Bob's a gigantic car. from the 1990s. I roll it down and I shout out to Huntsville, hey, just label them with the dye. No, you order them. You can pick what dye to label them. And different dyes will fluoresce at different wavelengths, okay? So I can choose to label 2 with a red dye and 2 with a blue dye. Now at this point, a blue dye doesn't really fluoresce blue because blue is higher energy. It's actually all of these colors are like within the yellow to red range, but we just sort of call them different colored dyes just to help differentiate them. It's confusing, but pretend I said nothing about it, okay? The camera is going to be able to differentiate this red dye and this blue dye. So if we tell the software, hey, red peaks are TPOX, blue peaks are VWA, okay, the camera's like, why are you talking to me? I'm an instrument. But we run this reaction, and now we still have 4 peaks. Does everybody see how this slide looks exactly the same as what we had before, except 2 of them are blue and 2 of them are red? We know because we ordered the primers. Oh, well, TPOX is red and VWA is blue. So when we go to our chart, there's no mystery. It's the 194 and the 206 go with TPOX. The 198 and the 202 go with VWA. All right, does everybody see that? You use a different dye, okay, and now it's very easy for the software to split these up and for you to know which one came from one loci and which one came from the other. All right. So ideally, if we want to amplify 3 loci, we use 3 different dyes. If we want to amplify 6 loci, we use 6 different dyes. If we want to amplify 16 loci at one time, we use 16 different dyes. The problem is we can't use 16 different dyes. Because in order for this red dye to be differentiated from the blue dye, the camera needs to be able to differentiate them. And the camera can't differentiate a red dye between a red-orange dye, between a orangey-ish, redder-ish dye. Like, remember, they're all sort of coming out of the same colors. They really need to be separate colors, and you can only squeeze so many colors next to one another. Right now, the maximum number of colors that they can get for a camera to differentiate them and not get confused is, I think, 7, okay? Because the old timers use 5, and then the new kits use 6, and I think some of the like newer kits are trying to use 7, but I think usually like 5 or 6 dyes is sort of standard as far as how many different dyes you use. But we want to analyze 16, so we need another approach. This second approach is a little bit more difficult to understand, okay? But what I want you guys to focus on, I'm going to rearrange some of my windows here so I can draw, okay? I want you to focus on the fact that, because we haven't really gone over this, like these are the repeats right here, okay? So this is 4 base pairs repeated 9 times. This is 4 base pairs repeated 10 times. This is 4 base pairs repeated 10 times, or excuse me, 12 times, 10 times, 4 base pairs repeated 11 times. Note that the primers don't attach right next to the repeat region, okay? Because the difference between, let's go down here, the difference between 10 and 11. The difference between 10 and 11 is going to be 4 base pairs, okay? If you put the primers right next to each other for 10 and 11, okay, then the 11 is going to be 4 base pairs bigger than the 10, okay? If you move the primers and put them all the way out here, okay, to amplify, even though the total fragment of DNA is going to be much larger, it's still going to be where the 11 is going to be 4 base pairs larger than the 12, or excuse me, than the 10. God, I can't even get my numbers right. All right. So just visual, when you order the primers to amplify an area of DNA, you can put your primers close to the area of DNA. It's going to get amplified. You're going to be able to measure it. Or you can put the primers farther away from the DNA, and you're still going to be able to amplify it. Okay, so how would this look? All right, well, before they were all coming out at the same time, right? 9 was going to come out, 12 is going to come out. Okay, for here, looking at these pieces of DNA, which allele is going to come out first? I want you guys to answer this in the chat box. The 9, the 12, the 10, or 11. Based on the fact that smaller DNA travels fast, what's going to come out first? Okay, and yeah, the 9 is going to come out first, right? Okay, now here's where it gets a little tricky. Which of these alleles is going to come out second? Which is going to come out after the 9? Because we know the smaller pieces of DNA travel faster, okay? So you may look and go, well, wait, the 10 is the, but no, we're looking for the total fragments of DNA. Remember, this area, the green, light green area that represents the repeat, okay, that's not going to affect the total size of the DNA. In this situation, can everybody see that even though this 10 allele has less repeats than the 12, based on where we put our primers, okay, the 12 is actually smaller. So in other words, all of the alleles from TPOXX are going to come out first, okay, and then all the alleles from VWA are going to come out next. Okay, what is that going to look like on a chart? Well, all of a sudden, we don't have the 4 base pairs that are coming right out on top of one another, where all 4 of those peaks are between 196 and 206. Okay, notice for VWA, we just have a different chart. Okay, notice that this is still, these are 4 base pairs different. We still get a chart going from 7 to 13. Okay, but because of where we put those primers, when we designed them and ordered the primers, we have created where all of the alleles for TPOXX are going to come out first, and the biggest allele for TPOXX will come out at 210, but the VWA alleles won't start coming out until 220. Okay, so they're not going to come out until later. Okay, so in other words, if we were to draw this electropherogram, oh, the software sees a peak 194, oh, a peak 206, okay, and then it waits a while, and then, oh, a peak for VWA and a peak for VWA, like this. And we know that all of our TPOXX alleles are going to come out in this range, and then all of our VWA, so it's still where we get 4 red peaks, but based on the timing of when they come out, we're able to differentiate between TPOXX and VWA. All right, so it's actually a combination of changing dyes and changing when they come out, where we can very easily multiplex for 16 loci. Okay, because picture this, picture we have 4 colors, okay, blue, green, yellow, which I don't show here, and red, okay? And picture, we can go down 186 to 210. We can have some coming out from 250. We can have some coming out from 250 to 300. So you can imagine if we had 4 colors going across and then spaced out so they come out at 4 different periods of time, we could very easily get to 16. 4 colors across, 4 down. And then no matter what peak came out, by looking at the color, we would know which loci it is, and then also by looking at the size. Okay, if it's a green 232 peak, there's no question that's D5S818, because that's what this chart is for. Okay, if it was a different color, it wouldn't be D5. If it was a different size, it wouldn't be D5. Okay, so this is what's actually happening. You have a whole bunch of different colored peaks coming out for a whole different and they don't come out just within this small range of when one locus would come out. They sort of come out over a long period of time. And notice, like, these blue peaks are from one locus, these blue peaks are from another, these blue peaks are from another, and these blue peaks are from another. They're all separate. And what the software does is it takes this somewhat confusing electropherogram and boom, this is what the software can do to it. These are not 4 separate samples. Okay, these are not 4 different runs of the same sample. This is, you took an amplification reaction that had 16 sets of primers in them. Some of the primers were labeled with a blue dye. Some of the primers were labeled with a green dye. Some of the primers were labeled with a yellow dye, which you can't see yellow, so that's why it's black. And some of the primers were labeled with a red dye. The company that sold us this kit with these 32 primers in it, 16 pairs of primers, 16 forward, 16 reverse primers in them, they designed them So D8 is somewhat close to the repeat regions. Okay, all the alleles for D8 are going to come out somewhere between 120 and 180. D21, they moved the primers a little bit farther apart, kept them blue dye. D21, they're all going to come out somewhere between 180 and 250. Okay, D7 between 250 and 300. CSF between 310 and 350. Okay, so as everybody see, by spreading this out, we can very easily get 16 loci that we are analyzing at once. And what the software does is normally all these colored peaks that would look like this, they're all coming out at the same time. But the software just splits it up and goes, okay, here are all the blue peaks. By the way, based on what you told me, all of the D8 peaks are going to come out under this bar. All the D21s are going to come out under here, okay? And you can very easily look at this, okay? What we haven't shown is right before we ran this sample, we ran a ladder. And we didn't have to run 16 different ladders. We run one ladder that has all of the alleles, all of the loci. What a ladder looks like, and I should probably throw it in here, is it will have a blue peak, a blue peak, a blue peak, a blue peak, and it has like 10 blue peaks that are between 120 and 170. Okay. And the peaks, remember, we have the tables there. Do I have this? Whoops. The tables as far as, 192 equals an 8. These gray bars here, this is actually representative of the table. What this is telling us is we ran the allelic ladder and it set all these gray bars for the day. And because, let's go ahead and erase this. Because, let's look at D21 here. Because this peak here is coming out in this gray bar, it knows, oh, that's a 29 allele. Because you just ran a ladder a couple samples before this that had a 29 peak from D21 in it. And this is where it came out. It came out exactly at 204. Okay. TH01, oh, this is a 7 peak. I know that because you just ran a ladder and the 7 allele for TH01 came out at 176. All right. So this is what it looks like. This is the data that you guys would be analyzing. You would just look at these peaks, and any idiot can do this. Okay. You just write down the pairs of numbers. Okay. At D8, oh, there's only one peak here. What does that mean? It's broken. They don't have a mother. No, it just means the mother and father are 12. So you write down 12, 12. Okay. You write down 29, 30, 8, 11. This is what your table looks like. Write down 11, 12, 10, 11, 9, 13, 11, 11, right? Compare your evidence to the data and you just see if they match. If they match, you do your statistics. If they don't match, then you return right in your report, excluded. This person was not the contributor for the blood at the crime scene. All right. So this is a little confusing. If you want to do the minimum amount of work, you should know what I'm looking for when I ask you, okay, how do forensic scientists differentiate alleles from different loci at the detector? Okay, just say you can use different colored dyes and you can move the primers so each locus comes out at different times. So you can arrange those so even if you label them all with a blue dye, you could get all the alleles for one locus to come out first, and then the second, then the third, and then the fourth. This is a little bit confusing. This specific section, like I said, is not in the book, but I think if you read in the book along with the previous lectures, it sort of helps visualizes what's going on. And that's really the important part, that you can somehow sit and visualize in your head, okay, after amplification, I got 10 million copies of this allele, 10 million copies of this allele. Okay, now I'm going to add some information. Okay, all of them are labeled. They all have a little fluorescent dye attached to them. So when they're all jumbled up, I just shove them into the beginning of the capillary. But as they travel through the capillary, all of those alleles are going to separate and move together, and all of the other millions of copies of alleles. And then they're going to hit the detector window. The detector can measure them by comparing them to the size standard. The detector can tell us what color it is. And then the end product is finally, we're here. We start with a blood drop on the wall. We've gone through this process, and what comes out are 13 pairs of numbers. Okay, so the exercise in the lab is really about giving you guys this information and having you copy down the 13 pairs of numbers, because when we do them in class, sometimes we can get messy electropherograms, we'll get like extra peaks, and you sort of have to be able to recognize the two big ones are the ones we care about. So I may switch to trying to give you guys some pictures that look like this, and then you guys essentially make the charts. And if you have the picture for, like, for example, blood evidence, and then I give you the pictures of the electropherograms for all the suspects, then when you make this chart, you'll find that the blood matches one of the suspects. All right, it's just a matter of, I need to figure out how the heck to do that without the proper software to do it. So I might have to go into Campus. Then again, that free software may allow me to take pictures, but it's not going to do the analysis. So anyhow, okay. Are there any questions? Sorry, I'm rambling. I forget. So in other words, plan on having class on Wednesday, but I might post an announcement on Canvas that says no class today. I'm still working on the assignment and I don't know what the heck I'm going to do. All right, well, that's it. I think I'm going to stop the recording. You guys are free to leave now, or you can also, I lost my thought mid-thought. Okay, so I'm going to stop the recording now. So if you guys want to ask any questions, you can do that either in the chat or you can turn on your microphone about the upcoming lectures, assignments, or anything else. We only got 2 more weeks to go.